Very good morning. I'm to Dr. Farah. My name is Sharani Rathakrishnan. My matrix number is 184202. So today I'm here to present my views and opinions regarding the question that the doctor have posted in Putra Blast. So I will start off with my first question, which is straight briefly on how you are going to develop the blockbuster drug. So when developing a blockbuster drug, there are several steps to be performed carefully in order to ensure that a new drug that can be turned into a marketable treatment for the patients. So the first and foremost step in the process of developing a blockbuster drug is choosing a suitable disease and also a molecule such as gene or protein that which is indeed involved in the disease in order for the drug to be targeted. So after selecting this selection can be done through a thorough literature search to identify the most prevalent health issues around worldwide, such as coronary heart disease, cancer, or diabetes. So after choosing suitable disease and also the possible drug targets, proceed to drug discovery. The first step that involved in the drug discovery is to develop a quick assay which to test the potential drug's effectiveness, such as the desired biological activity exerted by the drugs, and also it able to separate the bioactive compound from other inactive substances. For instance, like adding the compound to the tissue samples of the microorganism colonies to see the visible indication of the effective treatment. Next is the process of finding the lead compounds, that have some activities against the disease. It also act as a starting point for the further refinement. After identifying the lead compounds, we need to proceed to the optimization of the lead compounds, which involve the process of modifying the lead to enhance its potency and also to ensure that it exerts minimum side effects with the optimum therapeutic effect. And also it can be transported within the body to reach the site of the action. Last but not least, further experiments should be conducted to test the compound's toxicity and also its safety of the optimized lead through either through preclinical studies, which involves animals and test tubes or cell culture studies, and further proceed to the clinical trials, which involves various stages of testing the compound in healthy and also diseased humans. Next, I proceed to the second question, which is explain your strategies to make sure that the designed drugs can be become the most potent drugs. So a highly potent drug is actually the one that shows the therapeutic response with even at low concentration, and also it has a minimum side effects. The potency of the drug actually depends on two factors, which is efficacy and also the affinity, a term which is used to indicate how well the drug can be bind to the receptors. So the most straightforward approach to improve the potency or in order for the drug to become most potent is through appropriate chemical manipulation of the functional groups. So when a lead compound first discovered, it obviously it have a lack of potency, such as it may have issues such as unwanted or undesired side effects or poor pharmacokinetics. So these undesirable properties or features could be due to the specific functional groups that present within the molecules. So what we can do is like by modifying the molecules, in which removing the specific functional groups which is responsible for the undesirable effects, so will reduce side effects without losing the desired biological activity with the other group, which having the similar properties. This method is known as bioasteroid replacement. So this method should be employed in order to improve the or increase the potency of the design compound. In addition to that, the 3D shape of the molecules and the orientation of its functional groups also need to be considered, as we know that the properties of the drug is not solely depends on the presence of the functional group, but it also depends on the orientation of the functional group itself. So if the important functional groups which is responsible for the biological activity placed in the proper spatial arrangement or 3D dimensional arrangement, so that the drug can produce a strong interaction with the receptors. So this will eventually result in a strong therapeutic effect. So next is it is also important to consider the lipophilicity properties of the potential drug. We need to make sure the lipophilicity is in a correct proportion, because if a compound is too lipophilic, it can either insoluble in aqueous media such as gastrointestinal fluid or blood, or it may strongly bind to the plasma protein. Therefore, the free blood concentration will be too low to produce the desired effect. And conversely, if the compound is too polar, it may not be absorbed through the gut wall due to the lack of membrane solubility. Next, the bioavailability also need to be considered. For instance, like if a compound is with a very poor solubility, poor soluble aqueous solubility, it can also create a clinical problem. So in this case, the drug can be given as a prodrug, which is a substance, is a compound which is pharmacologically inactive, with a good bioavailability, which will convert into to active drug when it gets when the person is consumed or when it is administered into the body. So other than that, the dosage of the drug, the route of the administration, and also the interaction of the drug with the other compounds or substance also is very crucial to in order to determine the potency of the drug. So in a nutshell, the the molecules the drug molecules that have a good stability, but also the reasonable properties of the absorption into bloodstream, distribution, binding with the tissues, its metabolisms, excretion, low toxicity, and also the reasonable cost of production is important in order to make a most potent drug. So that's all from me, doctor. Thank you.